3D density maps is a powerful tool for studying sulcal imprint variation on extant human endocasts. The question I'm sure we would all like to answer is, how did the human brain evolve? According to Ralph Holloway, the evolution of the human brain has been an intermingled composite of allometric and non-allometric increases of brain volume and reorganizational events, such as the reduction of the primary visual cortex and a relative increase in both posterior association and prefrontal cortex, as well as an increase in cerebral asymmetries. To study this, we can look at genetic evidence if technology allows us, but also at the histological and gross anatomical features of the brain. In the fossil record, due to lack of preservation of the brain, we have to rely on cranial endocasts. Traditionally, two types of endocasts exist: the natural endocasts and the synthetic endocasts. Modern imaging technology has provided us with an alternative to extract more detail from some of the most fragmented samples. Previously, we used a modern imaging technique to document and create an atlas of sulcal imprints on modern human endocasts with great success. However, quantifying and visualizing the high inter-individual variation still poses a great challenge. For this, we had to look at approaches that are being used in the medical industry. We used one of the population-based atlas techniques, the density-based approach, to characterize the variation of sulcal imprints on an average endocast template. Our method runs as follows: in green to create an average endocast from our sample, and then in orange to deform all the imprints detected from our previous study to the common template and create a density map for each sulcus. Some of the results we got from this: clear distribution patterns in red can be observed for the superior frontal sulcus, the middle frontal sulcus, and the inferior frontal sulcus. Moving on, higher variation could be observed with more erratic patterns for the precentral, central, and postcentral sulci. Clear distributions were visualized for the anterohorizontal ramus and ascending ramus of the sylvian fissure, with a few outliers on the left hemisphere. Clear rostral distributions was identified for the temporal sulcus, with some outliers towards the posterior end. Finally, viewing from posterior, we had a clear red distribution for most occipital sulci, especially the lunate sulcus, the inferior occipital sulcus, and the lateral calcarine sulcus. Overlapping of imprints was observed when viewing the imprints on the frontal lobe, as well as laterally the precentral, central, and postcentral sulci, with more severe overlapping towards the superior medial border of the hemisphere. Major overlapping was observed in the sulci bordering the Broca's cap, as well as sulci of the occipital lobe involving the lunate sulcus. Just a few thoughts. This approach provides an innovative, non-invasive, statistically modeled method from which to reliably infer human endocranial sulcal imprints. For the first time, we could observe topographic overlap in sulci that is critically discussed in paleoscience, especially when we look at the overlap of sulci on the occipital lobe, which involves the lunate sulcus, as well as sulci in the Broca's area. One should definitely take caution when identifying these sulci on endocasts. Nonetheless, our analytical protocol afforded an objective perspective to contribute towards more refined endocranial research in paleoneurology. Expansion of our specimen database is expected to further improve the statistical power of observed features, thereby detect with improved certainty the observable remodeling of these cortical features, and consequently contribute towards a completely observer-independent approach and discussion surrounding the fossil classification and the contentious hypothesis of the evolution of cognitive abilities amongst hominids. I would just like to thank everyone that contributed to this project, as well as my sponsors and especially the South African-France Joint Research Program. Thank you.